Hello, everyone. Welcome again. Thanks for turning out. We've got quite a good turnout this evening. It's a miserable night, so well done, everyone. And welcome to everyone online as well. Hope you all have a nice evening. Professor Richard Friend gives his apologies for not being here. He's buzzing around the world again, as usual. We've got two speakers tonight. The first one is one of our student award winners, Filip Boskovic from the Department of Physics, the Biological and Soft Sciences group. And he's going to talk about, I haven't got the title yet, but RNA Rosetta Stone, folding RNA with the origami technique for pathogen diagnostics, which is a new approach to the identification of pathogens. Come on up, Filip.
Thank you, John, very much for introduction, and thank you everyone for showing up. So I will tell you a bit about a story today. So you're free to ask after if you have very specific questions. But what we are working on and what I'm working on is basically origami, but not paper origami, but you will see DNA and RNA origami. But first, the outline of this short talk would be we'll talk about the pathogen types, what pathogens can be. Then we will talk about the pathogen diagnostics, problems that we face today with, as well as how we can use a molecule that's called an RNA as a pathogen fingerprint, and how we combine two techniques that we use in the Department of Physics. One is origami and one are nanopores. And finally, what we can achieve with it. So when we talk about the pathogens, most likely you probably heard about bacteria and viruses, particularly the COVID-19. I think we are now very familiar with the viruses. And the third one also, you have also this malaria, so more eukaryotes, we call them, so parasites, fungi, and so on. And in this talk today, I will focus on some research that we have in these two papers that's actually focusing on the bacteria and viruses. When we talk about the golden standard, one of these you probably heard of, it's PCR. Another one that's more traditional are bacterial cultures, or growing the pathogen that you want. So cultures are usually very slow, so you need to wait a day or multiple days. And probably the PCR obviously is that you are limited to testing one pathogen at a time, maybe two. But we have in this room 1,000 pathogens that have very similar symptoms, because all of us are kind of carrier of infections. But the question that you would ask, you see a lot of in the news all around, oh, new diagnostic. But the point is, what would be an ideal diagnostic tool? First, we would like to access if that organism is viable. Is it alive? For example, if you have a positive PCR, it doesn't mean that you have infection, because the DNA that we detect or the molecule that you want to detect might be just there, but organism is dead. Then the speed, you want to be in at least a few hours. Then you want to detect at least one. I think I would say that's obvious, but in most of the cases, you are detecting tens or hundreds. And usually you want to detect variants, as it's also with the SARS-CoV-2, but also with the bacteria. It's very important to know the variant, particularly for bacteria, in order to give a proper antibiotic therapy. And also we would like to detect, let's say, hundreds of pathogens within the same test and tell which one do you have. And which pathogen do you really have? I actually have a personal story. That's how we got involved in this project during the pandemics. Its hardest question is this one. So probably you had infection, you went to a doctor and gave you antibiotics. You had, oh, I don't know, just in case. And I actually had a long time ago, because I'm allergic to most of antibiotics. So infections for me are deadly or not. And this is me after my first quarantine in 1999 or maybe 2000, when I was like 1 month in infectious clinic, and they were trying to do PCR tests and discover which virus I have. And obviously they couldn't, I think they couldn't, but I was lucky to survive after continuous fever for weeks. But 30% of early childhood deaths are actually directly caused by only respiratory infections. So we are talking still about the big problem. And that's area that we focus on, not early diagnostic or something like that, but we have a sick person in the hospital and we want to give him, her, or them treatment. And our solution was a bit, let's say, so Stephen Baker gave a talk a few weeks ago, our collaborator. And when I told him this idea, he was like very speculative about it, saying that, oh, this might not be possible. We wanted to use a ribosome, which is our machinery in the cell that produces the proteins, basically not to use a ribosome, but the consistent part of it, which is RNA, that we call ribosomal RNA. And the reasons are very simple. So tree of life, how we divide all organisms on this planet, how we determine that this is Escherichia, this is Salmonella, this is human, this is bacteria, is using ribosomal DNA. So it's a gene that codes for the ribosomal RNA. Second one is sensitivity. In our cell, we might have one gene or few genes for ribosomal DNA, but we have tens of thousands or even 10 million ribosomes per cell. So if you look in the number 1 versus 1 million is definitely a big difference, like orders of magnitude. So we are trying to focus actually detecting these abundant molecules. And the beauty about RNA, also with the vaccines, that's why we kept them on -70, like Pfizer and Moderna, because they're very degradable. So they degrade on the room temperatures within even a day. So this was a good thing because it can tell us if the bacteria is alive. If it's alive, it has ribosomal RNA. If it's dead, there is no, it's only DNA. But the question would be, how can we detect actually ribosomal RNA? And that's where we jumped in with this origami technology, where we fold actually here the molecules. We don't fold the paper. So we have a single-stranded DNA or RNA. So in this case, it's RNA. And you probably know the double helix of DNA. So it has 2 strands. So here we have only one. RNA is one-stranded molecule. Then we design and added this in a gray, shown in a gray. So these DNA single strands. And after that, we create kind of this strange hybrid. So it's a duplex made of RNA and DNA. And then you might see or might notice in certain positions, like 3, 1, 1, 3, we can put a structure of different size. So if we want a big structure, let's say it can be 3, or if we want a small structure, it can be 1. And that's also nice. So in that way, we give a molecule with unknown features, very simple readout, physical readout. But to read that design, we need a method. We need somehow to have a fast readout. And to do so, we use actually electronic measurement, and they are called nanopores. So nanopore, saying in common words, is just a very tiny hole through which we apply actually electric, apply bias or electric field. And as you can see here in this cartoon, is basically by having a smaller constriction, you increase resistance, but you also, if something goes through that hole, you can see it. And here is example. So here on the left, you can see the nanopore. This is just a hole. And now we will actually drag the molecule towards the positive electrode. And on the right, we will see the readout of it. So when we pass it through, what happens is that the current drops. Why it drops? Because we block the pores. It's like you cut a cable in half. The current will not go up. And also, each of these labels that we put, or these structures, we can read. So every single pathogen, we can assign a certain code. And then I was wondering what would be the maximum number of codes that we can create. And we showed that we can create 10 different levels with the 10 sides. If you do simple math, we basically can detect 10 billion different pathogens. So it's more than existing in the planet. But also this is applicable to biology. We can detect 10 billion different RNAs, if you would like, with very good accuracy. So even by decreasing this density by order of 5, magnitude of 5, it should still be fine. And with this origami story, it goes more to what we got so far. So how the workflow does, we take a patient sample, we basically boil it with these DNA oligos, so these DNA strands. And in that same time, we break the cell. So we isolate RNA in the sense and create these codes. Then we just remove this debris from the cells. take this upper phase, which we call supernatant, and finally we just put it through these nanopore sensors and basically have kind of 30-minute workflow. What's really cool about this, compared to vaccines that degrade really fast, you will say, oh, but you're boiling, it's probably chemically would even destroy it. We discovered that creating this coats actually stabilize RNA, which is also important for the vaccines, but also enables that these can be done without freezer or any external sources. And even more by actually not purifying. So with the COVID test, when you do PCR, you wait a few hours because someone needs to wait for this reaction to happen. But here we don't have a reaction that needs hours, it needs 5 minutes. So we can really speed up the process. And then we were wondering just what was available around. Can we detect in different molecules? So this is even the species identification, where we actually detect here the human RNA, ribosomal RNA, rRNA stands for ribosomal RNA, mice, Escherichia coli, even Salmonella, that's a very known pathogen. And just a reminder from what we said before, so there are hundreds and even millions of ribosomal molecules. And these, for each of these, we can create a code for human, let's say 111, and then for the, I don't know, Salmonella, it can be 3113. And in that sense, we can multiplexly test for multiple pathogens in the same test. And for PCR, you would need probably half of lung to do 100 of the tests. But here we use the same amount of samples. When we talk about specificity, Salmonella is a very good example. It has thousands of different variants that have a different susceptibility to antimicrobials. And for that, knowing which variant you have means that you will get appropriate therapy. And here we just an example, we show that we can discriminate 2 of the variants, Tifimurium, that's one very known for the animals as well, so it both attacks animals and human, and Newport, which is actually the name of variant given by Newport City, if someone is around from it in the United Kingdom. And then we were also wondering what can happen, not only that you have a pathogen, but if you have one of you maybe recently had respiratory infections, it's very likely that you have a co-infections, meaning that you might have a multiple pathogens infecting you, one more or less. That has got very known in the late stages of COVID when the flu and different viruses kind of combine with the COVID, but we couldn't determine. COVID test gives you answer for one pathogen, but we are here looking answers for all of them. And we actually, this is one example where we actually use a bacteria and these 2 viruses that infect that bacteria, and we can actually identify bacteria, which is on the end here, one virus, another virus that infected them, and even quantify it. So we can say which virus is probably predominantly infecting you. And to summarize this is we show that we can, using ribosomal RNA, detect viability of the cells, that we have really a good speed for a test with almost 1 bacterial sensitivity, but without using this PCR reaction, so making more copies. We show that we can detect different variants, even test for hundreds of bacteria and viruses in parallel, and even do co-infections. With that, I would like to thank our collaborators, Stephen, that you had the chance to hear a few weeks ago, and Professor Mike Howard, also here from Cambridge, and my supervisor, Ulrich Kaiser, and my group that we've actually worked on these studies. Now we have also the already 2 years spin-off company from our lab and university, kind of bringing that to you. And with that, I would like to thank you and open to questions.
Filip, that was fantastic. Are there any questions in the hall? Yes, here we go.
Yeah, in the stage of debris removal, do you use filters or a centrifuge or combination?
So we do use, so I will go back to the workflow. It would be easier to show you. So here, basically what we don't use any chemical isolation. So you don't need expensive chemicals. You're not limited by proteins. So if you know the majority of the problem with PCR tests, you need the proteins. Someone needs to make those proteins. Here, everything is basically linked to the simple physical principles. And with the physics and electronic measurement, here we can maybe need the centrifuge, but it's not a mandatory step. The key step was the one that I circled here, actually trying to get RNA that's clean enough from the cell without actually using complex biochemistry, I would say.
Thank you. I was just wondering what the numbers stood for, like on the code, the 1s and the 3s and the 2s. What did they represent on the molecule?
Sorry. Okay. Numbers on you mean on these barcodes?
Yes.
So the number represents higher the number, we have a bigger structure. So in nanopore, because it's a resistant method, basically, let's imagine this is a cable. So if we cut the cable in half, you have a drop of the electrons that can go through. Here, basically what we detect is a ionic current. So if you have a bigger number or bigger structure, you will have a higher drop. That's why, for example, here we can see, maybe this is a good example. We have 1, 1, 3, 1. So 3 is the largest, that's why it's deepest, because it blocks the most of current.
I've got an online question here from Francis Skeffington. Is this method in clinical use?
No, that's a good.
Why not?
Yeah. So of course, particularly the pathogen or infection diagnostic field is not the one that gives you most money, because usually, except the COVID, that's not a good example. We usually, if you go to the doctor, he doesn't do tests. He even doesn't do antibiogram. He just give you antibiotic and send you home. So of course, the market UK is not ideal. But what we are more focusing on, this method is kind of already on the market with our collaborators, our collaborator company, just under a different name. But we are able to detect actually RNA for other applications. This is infectious disease, but we can do for probably cancer and any other actually disease or infection that you would like.
Absolutely brilliant. 2 questions. One is, when you say sample, what is the sample? Is it just like some blood, or is it being treated in some way? And the second question is, you saying that you assigned these IDs. I noticed that the same ID number seemed to be used for a number of different variants. So I didn't quite follow how that ID number identifies your 10 million different things.
Mm-hmm. So for the patient sample, that could be a lot of things. It just needs to contain cells. For Salmonella, what we use here are cultures. So it's a growth bacteria in the lab. And in one of the papers we had in the Nature Nanotechnology, we actually use the patient swabs. So the same. So our collaborators did the PCR test, and we stole a bit of their samples and actually did our test. So this just shows that without even changing the way how you prepare the samples, we can just fit in, which is usually good. And answer the second question, so each of these codes has a different number of bits. So as in coding, you have 0, 1, and then if you want to code letter A, I don't know, you have 0, 0, 0, 0, 1, 1. Here we actually also do that. Just we have 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in that sense, we have much more wider opportunity to use it for variants, unique sequence. So every variant, every organism has a unique order of letters in its RNA. And we use that unique order of letters to design unique label for it. For example, if you look, Salmonella and Escherichia coli are very close relatives. So they are shown here on the bottom. But actually, we can very quickly even use the same one. So the last red is the same, I think, as the third here. So basically, we can use the differences to discriminate between them.
One more question, I think. There's one.
Thank you. I'd be interested to know the sample that's got into the sample has got into the lab. Now the scientist is now going to work on it. what is the time lapse from start of receiving the sample to actual identification? And from that point to do the report that's necessary after that?
Yeah, so the part before and after we haven't done, so I can't estimate. The part when we get the sample, let's say swab sample, to the point where I can give you a result, that's around half an hour to 1 hour. Including this report and before that part we are not doing as a scientist, but that's something I think more relevant to someone who's actually doing that in practice. But I presume that would be standard. So we are comparing to the standard method. These two parts are almost identical because here computer tells you this is likelihood that this is present. We are very certain that this is this pathogen. And then the doctor can just read and say, oh, you have this pathogen. So I'm not a GP, but I think that I would say would be straightforward.
Okay, we'd better call a halt there. Thank you very much, Filip. It was
Thank you.
fantastic.